Okay, well, we'll get started. Welcome everybody to the first post-Thanksgiving IMAG MSM working group mini seminar on virus modeling and immune modeling. We have two great speakers lined up for today, Jacob Barhak and Gary Ann. As usual, I need to tell you that the meeting will be streamed and recorded and will be made available. So please don't say anything you don't want to have in the archives. Our first speaker for about 15 minutes is Jacob Barhak. He's going to talk about reference model accumulates knowledge with human interpretation. Jacob, are you ready to queue up? I see you there.
Yeah, I will. Yeah, okay. I will also add some information for everyone. This is how you can get it. So I will add some more data over there little by little because it takes a while. You can find in the chat some references for later on that I'm going to talk about. But for now, oops. Yeah, and I'm just want you to have those available. So you can browse this information, and some of it is already related to IMAG. So I wanted you to have all those links. Let me share my screen. And what you're seeing here is the presentation. It's the first link I sent you. It was supposed to be a poster for the last IMAG meeting, which was delayed by the pandemic. It was supposed to be about the diabetes model and the new inventions there, but now it's actually applicable for COVID, and I'm going to show it near the end. I'm talking about the reference model for disease progression, and the reference model started because I saw the models for diabetes, and I saw that you need to be able to combine multiple of those together, some sort of an ensemble model. So in 2012, I started developing it. What you see here is the diagram of the diabetes model. It's a state transition model, but notice the states are in different processes and the processes compete. Let me explain. If a person dies out of a heart disease, MI, they cannot die out of a stroke afterwards, and vice versa. Also, if they get hit by a bus, which is a competing death, or by another disease, then they cannot die from those conditions, and other conditions cannot develop. So this is a race that's coming here, and the model is able to do this and compute it because it runs micro simulation, very similar to agent-based simulation, and it runs for each person. It stochastically runs what might happen to it to that person using Monte Carlo techniques. However, you don't do this for one person. You do this for many people and for multiple populations of multiple populations. Each one has many people inside it. And the information is heterogeneous because people are different. And when you run this for a model, you need a lot of computing power if you do it with many people. But I actually need more computing power because I'm not running it for one model. Each one of the transitions you see here in this diagram can be governed by a different equation, and this equation can depend on other states that are in other processes. So there's a mix of things that are happening, and this gets governed by multiple equations that kind of interact with each other, and they all together become an ensemble. I started as these equations competing against each other, but later on it became an ensemble of those. Now, having an ensemble is one thing, but what do you do with it? Well, the reference model is called a reference model because it has references that you have to adhere to. First of all, the models, where they come from, and the models are kind of assumptions. But then from the other perspective, you have something that you need to compare the simulation results for. So I take clinical trials that were done for diabetes, and I compare the model that was executed on a specific clinical trial population and compare it to the end result of the clinical trial. And I create a reference where I can actually later on show you how well or how unwell each model behaved. This technology is patented. So unlike many of you, I'm not academic. I left academia to pursue this, and the last 8 years developed this technology. This technology is now patented. So if you're doing or need to do something similar, talk to me. I also have a conflict of interest because I'm not an academic and I want to declare it. It's important for you to know. I contract with other companies to do the work. So therefore, whatever I'm showing you may be influenced by other things, but I try to be as just as I can. You'll find all sorts of information about what I do on my website, and as transparent as I can be. And also you'll find in my publications a lot longer, long conflict of interest declaration. Go back to the reference model itself. In 2012, I started it. Each year I added pieces of technology that enhanced it a little today. So in 2012, it started. 2013, I added cloud computing and high performance computing. You need a lot of computing power to run all those models against all those populations, especially in micro simulation level. I added evolutionary computation to better mimic the populations I'm modeling. And then object-oriented, because I had so many populations, you had to organize it. I added the assumption engine. Basically, this is the engine that drives the ensemble model. In 2016. What you're seeing here is the number of models or model combinations that I was running. So you can see at this point, it's infinite number of models. Basically, if you have a set of models and you have a modeling space, it kind of becomes infinite and you need an ensemble to resolve it. Because, although the basis functions there are only a few of those, when you combine them, they become infinite number of combinations in an ensemble. 2017, I connected to clinicaltrials.gov. This is why you see the jump here. This is the number of validation points I'm validating against. So 2000, it get to about 360. I started from about 150. This is where I connected to clinicaltrials.gov. You see the jump, but also I continued this, added some graphics, and this year I added human interpretation, which I'm going to talk about in a second. Today I'm at about almost 500 validations. This is the number of populations I was modeling, and you can see this is number of populations, the cohorts, and again, the validations. And you can see how this number grew throughout the years. Now, after I have an interface to clinicaltrials.gov, it's much, much easier. Now, let's talk about the equations. Notice there are many types of equations that on anything that can affect diabetes, how biomarkers change, how there are multiple death equations, and multiple types of death equations as well. MI equations, stroke equations, the number of those, I stacked up all the equations and put them together. This is the 2012 model. And you can see how the number has risen throughout the years. This addition of interpretations, I added 6 interpretations of so-called experts. Those are not real people. Those are, think about it, a person who would think like this is considered an expert. And I added 6 of those. And I want to focus exactly what I mean here, because this is the new part that I was talking about. I want to explain. So when you conduct a clinical trial, you eventually have reached some endpoints, and those endpoints are either single endpoints or composite endpoints. Let's say if a person has an MI or heart attack, it's usually a single endpoint. If they reached it, then they count it as one if they reached that condition. However, you can combine them. So for example, cardiovascular disease is combined from MI, stroke, and some other conditions. Those definitions, even within trials that are conducted in different times or different places, or even the same environments, are sometimes different. So the numbers reported in the clinical trial represent slightly different measurements. Ideally, those will be reported as ICD codes. However, not all trial reporters and ICD codes look at clinical trial publications. It's becoming better, but it's not there yet. And even if there are ICD codes, the ICD codes not always match between the definitions of the trials. And of course, there are multiple levels of multiple versions of ICD codes, and you need conversions, and things get lost in conversion. So eventually, you need a human to look at those numbers and tell you what those numbers mean. Are they overestimated compared to what your model is calculating, underestimating? Also, one more thing, some definitions change. I gave sepsis as an example, but sepsis definition has changed throughout the years. Gary Ann is after me. I saw a lot of paper on sepsis that he wrote, so he will confirm it. Sepsis has changed throughout the years, the definition of sepsis. Same thing happens with other things. So if you have older clinical trials and newer clinical trials, you need some level of interpretation, and some study actually are aware of it. So when they publish the study at the end, and I'm giving the example of the record study, they actually tell you if we count our endpoints like this, or we count them in a different way, then the numbers we get are slightly different. And they publish this. This is part of the publications. They are honest about that, which is good. So if my model counts an MI according to one definition, and especially if an ensemble of models, that they can come from different definitions, when you combine them together, you eventually need something to, some filter to, before you do the comparison to the observed data from the clinical trials, you need some buffer, and this is human interpretation. And I'm adding it this year, just like the models, you have multiple human interpretations. Think about the situation in COVID time. Even now, they report numbers of infections per state or numbers of deaths per state. Are those numbers the same per each state? So first of all, there are errors that they find every once in a while and correct. There's all sorts of anecdotes about that. But beyond that, how did they reach the infection number? Did they is it a percentage of the number of people they tested? And did they test everyone in a proper way, or is it only the number of positives that they found in their test, which maybe the test sample is smaller than the population? All of those questions are relevant, and you need an expert to come in the middle and says, I believe those numbers, or I think those numbers are overestimated or underestimated. Same thing with death. If someone had COVID the minute they die, did they die out of this disease, or maybe they just died of something else, but they said that they had the virus and therefore they were registered as COVID? So those are questions that an expert can give you some interpretation. Let me show you how this works. The reference model actually takes all of those equations that I discussed before and gives them a weight at the beginning. At the beginning, you believe every equation that you gave and each interpretation of each expert the same way. This is why you see those bars with a certain color, the same height. This is the influence of each model. You can see the MI models, stroke models, also mortality models, and human interpretations over here in cyan. What you see here in the upper left graph is the clinical trials. This is like acronyms of that represent the clinical trials and the cohorts. So you can see the cohorts over here. There are statistics about what's the average blood pressure, how many were smoking, and so on and so forth. You can play with the display a little bit, but this is less interesting for now. It's more important to explain what the heights are. What you see, the height of the circle, it is the error between the model prediction, the ensemble model, and the actual observed results of the clinical trial after interpretation. So here you're seeing the combined interpretation, but let's look at expert number one. Expert number one in my situation here says, okay, I believe all the numbers reported are defined exactly like the model. You don't need to add any buffer in the middle. No human interpretation is needed. This is what the model therefore would see. The model will try to optimize, and what you see here is the convergent curve. We'll try to optimize this, and you can see all of those, each iteration, and each one of those iterations takes a long time. Even on high performance computing machines, each iteration you'll see some sort of fluctuation and improvement, gradual improvement towards convergence. And in the bottom, you see how all of those bars change height, and you can see some of the bars disappear. Those are models that will not be used by the ensemble. The rest of the models some of them become dominant and say, okay, this model represents well with all other models and other interpretations represent the best what's going on in this situation, the question that you ask the machine, the query that you ask the machine. However, notice that different experts will see the same the results differently. And this is something that we can observe and look at around when we change the expert. However, the combined experts is the one that takes those experts in those weights, believes them this way. So for example, the most believable expert is expert 6. He's about 50% of his prediction is used, about 40% from expert number 4, and about 50% from the last one, which is number 2. The last 3, the system tells you probably ignore them because the information they provided doesn't match well. So the same thing that I applied to diabetes can now be applied to COVID. This is a recent publication a few months ago about the initial use case. You can find it in Curas. I sent you the link. This is the diabetes model. This is how the initial model for COVID, this was how it's published there. For COVID, I'm doing something very similar, but I'm doing it for US states, not for clinical trials. And I combine the infections and deaths, and the fitness score here shows the difference between infections and death per state. When I run the model, and here you see the convergence on the bottom, you can see the convergence plot, and on top you've seen some of those outliers drop down to the ground. This means the model got optimized, and what I'm optimizing is parameters. The model here is very, 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 very simple, very similar to an SIR model with a touch of ensemble, just for the mortality. I have 2 mortality models there, both derived from the CDC, high and low, because this was my initial use case. Now I'm working on a more advanced model. Maybe it's worthwhile saying that the model results that you've seen here, those are computed, just to give you an idea, 9 hours on the Rescale cloud. Rescale gave their infrastructure because of the pandemic. They donated their resources. Microsoft Azure gave money, so I was able to run it. It takes 9 hours for 880 cores and 20 machines on the Rescale cloud to compute that. So it's a lot of computations. This is now a model that John Weiss is here. This model is currently running on a server that he hosts today. And also I want to thank Jared Hodges for taking care of the simulation there. And the model here is the current COVID model. These are not final results. This is just to show you the flavor, because I want to ask you for help from the working groups. I actually do need help from some of you for some things. What you're seeing here is the same idea as before. We have multiple models though, however. The reds represent infection models. For example, infectiousness curve. This is why I asked Alan Perelson for a few weeks ago. He gave a talk here in this working group, and I started communicating with him. Maybe I will able to plug as an infectiousness curve in one of those infectious models. If you have more of those infectious models, please do contact me. I'm interested in plugging them in. There are transmission models here. How do the models transmit? What you see here are response models, how people respond to the pandemic, how they behave. Those are behavioral models, the cyan, and the rest are mortality models. But more importantly, I currently don't have experts that will interpret the numbers from the US data. I'm showing here every 5 days the numbers that come from the COVID tracking project. And my last publication, this publication, both take data from the COVID tracking project, so I want to thank them as well. At this point, I am looking for your help. I will stop the talking and I will ask you for questions, and hopefully you have questions that say I can help.
Great, thank you very much, Jacob. I think that your focus on aggregating information from different sources and connecting to clinically meaningful results is important. We have time. We have to keep moving a little bit so Gary can speak, and we have a little time for discussion. But do we have time for one question or two questions in the room?
So let me just ask Jacob, because you sent an email to us. So what exactly do you want? What sort of community engagement in terms of it's because it sounds like you have data, So do you need these kinds of population level or anything? I'm just trying to figure out what it is that you need
Okay.
input on.
So population level, the population comes, the nice thing about a reference model doesn't need the individual data. It can live out of data that is per population statistics. So I have the ability to merge those statistics. So I'm not looking for the population statistics unless we want to add more populations for different countries, which is a possibility. Some people talk to me about this, but from a research perspective, it's interesting to compare different countries or different behaviors, but I'm more focused currently within this working group on things that people actually develop. So for example, when Alan Perelson talked about, I think it was 3 weeks ago or a month ago, he gave a talk about how he has the he has the infectiousness curve within the upper respiratory system and lower respiratory system, and how much virus is there, the viral load. When he talked about those, I sent him an email and he tells me, oh, here's a paper, and he actually has an infectiousness curve equation there. Says, oh, the person is infectious after transmission this much. And he has several things there. So I'm trying to incorporate this equation. I'll have to talk to him more, but this is one example of things I'm looking at now. And I know some people already send me something similar. I need some of those that I can actually plug in, and then those assumptions go into the model. The model will compose those together and tell you which one fits better the data that you have. And the data can come multiple sources. The other part is the human interpretation. If someone looks at all of those per state numbers like deaths and infections and can tell you this is overestimated, underestimated, this is data that I can use to plug into the model as an interpretation. It's a human interpretation thing. It doesn't have to be correct. It has to be on human intuition, or maybe sometimes it's human, it's what human thinks, and sometimes it's right, sometimes it's not so right. But the more of these I have, the better. And if you have other types of models, we can talk. Maybe they can fit as well. The ensemble model just accumulates information from multiple sources. Did I answer?
Yes, thanks, Jacob.
Okay, thank you, Jacob. I think we better move on. I hope people can talk offline, have fruitful discussion. Thank you again, Jacob, for being willing to speak. Now we'll move on to Gary